Okay, in this lecture, we will walk through what happens to a lipid nanoparticle in the body. We will start with how it's made, then we will follow it through the bloodstream, and then into cells, and then ultimately the biological effect. So, the key idea is that the rules change along the way. A design choice that helps at one stage can cause problems later. So this lecture is essentially about understanding these transitions. So at each scale, I know maybe molecular, particle, cellular, tissue, and organism. So different physical and biological constraints come into play. And these things shape delivery performance. So in this lecture, we will highlight key limiting phenomena. That includes protein corona formation in circulation, endosomal escape, and the trade-offs introduced by pegylation. Finally, we will look at how molecular scale properties propagate upward to influence organism level efficacy and safety. This slide gives a high level overview of the LNP journey. We can think of delivery as a sequence of stages: formulation, circulation, cell entry, endosomal escape, RNA translation, and eventual clearance. So each stage operates at a different biological scale, spanning particles and tissues, cells, molecules, and the whole organism, right? Importantly, the constraints at these scales are not independent, they are coupled. So properties defined early in the process can propagate forward and shape what happens downstream. A simple example is lipid pKa, a molecular property that strongly influences endosomal escape at the cellular level, which in turn affects efficacy and tolerability at the organism level. So this coupling across scales is what makes LNP delivery fundamentally a multi-scale problem. So let's start at the particle scale with the manufacturing and assembly. So the objective is straightforward. We need to make a uniform, stable particles that efficiently encapsulates RNA. But in real life, this step is not easy because particle formation happens very quickly during mixing. Small changes in formulation or flow can push the system um, down very different assembly paths. As a result, outcomes are highly sensitive to things like lipid composition, nitrogen to phosphorus ratio, mixing geometry, and the total flow rate. A key point is that the internal structure of LNP is not something we can easily regulate. It emerges during assembly and reflects how the process was run. So that's why manufacturing is not just a production detail. It is one of the main drivers of particle level properties. Because assembly is so sensitive, a few things tend to go wrong during manufacturing. You can see aggregation, you can see particles drifting in size, you can lose encapsulation, and you can end up with particles that look similar on the outside but not so internally. Small changes in formulation or process conditions can push the system in the wrong direction. A good example is PEG-lipid. Increasing PEG by just a few mole percent can reduce RNA encapsulation, even though everything else looks unchanged. So that's why parameters that seem minor can end up dominating particle behavior in practice. So when it comes to step two, once the LNP enters the bloodstream, the proteins in the blood quickly stick to the particle surface. So this forms a protein corona and changes how the particle behaves in the body. So the corona has a big impact on where the particle goes and which cells it interacts with. At the same time, the LNP is being cleared by the phagocyte system. In on-pattern case, this works in our favor. So the particle picks up ApoE and is taken up by the liver. For many other applications, the same behavior becomes a problem. PEG plays a central role here. PEG helps the particles survive in the bloodstream, but too much of it makes it harder for the particle to interact with cell membranes and enter target cells. So, circulation is always a trade off between staying in the blood and being able to deliver cargo to the right cells. At the cellular scale, delivery runs into two main problems. First, lipid nanoparticles cannot cross the cell membrane on their own. So cells take them up by endocytosis, which traps the particles inside endosomes. And the second problem is endosomal escape. So this is the main bottleneck in RNA delivery. Only a small fraction of particles ever make it out into the cytosol. So most are sent to lysosomes and broken down. Ionizable lipids are designed to help with this step. So they stay mostly neutral in the blood, but they become charged once the environment turns acidic inside the endosome. The change in charge helps the particle interact with the endosomal membrane and promotes escape. Because of this, small changes in lipid chemistry can have a large impact on overall delivery and efficiency. Once the RNA reaches the cytosol, delivery is still not finished. So free mRNA has to survive long enough to be translated. RNases degrade exposed RNA very quickly, so stability immediately becomes a limiting factor. At the same time, unmodified RNA can trigger innate immune responses like RIG1 and MDF5. When that happens, translation drops and inflammatory pathways turn on. Even if the RNA stays intact, it still has to engage ribosomes efficiently to produce protein. So that's why RNA sequence design and chemical modifications are important, and of course largely determine protein expression. So now we are at the organism level. Here, our main question is, do we achieve high efficacy with acceptable safety? For efficacy, the readout depends on the payload. For mRNA therapies, the question is whether enough functional protein is produced in the right tissues. And then for siRNA, it is whether the target gene is sufficiently and durably silenced. On the safety side, accumulation of lipid components can drive liver burden and hepatotoxicity. Very high levels of protein expression can contribute to inflammatory responses, and both the RNA payload and other components can activate innate immune pathways. Physicochemical properties of the lipid components, such as effective hydrophobicity, ionization behavior, and the lipid tail structure, strongly influence biodistribution, tissue exposure, and liver burden. So, at this final layer, we are no longer talking about individual molecules or particles, but about how all upstream design choices express themselves in vivo. Okay, so now this brings us to what is that people call as the coupled effects problem. In LNP systems, changes at one scale affect behavior at other scales. These effects are causal, not independent. For example, changing the ionizable lipids pKa or tail structure changes its charge state and packing behavior during formulation. So that packing behavior influences the particle's internal morphology, and particle morphology affects the probability of endosomal escape. Endosomal escape then determines how RNA reaches the cytosol and is translated. Those translation levels ultimately determine organism level outcomes such as efficacy and toxicity. So the key point is that none of these layers can be modeled independently. Optimizing performance at one scale, such as high RNA encapsulation, does not guarantee success at another scale, such as endosomal escape. For this reason, single scale models are insufficient. Predicting LNP behavior requires integrating features across molecular, particle, cellular, and organism scales. So, to summarize, LNP delivery is controlled by variables at multiple scales. At the molecular level, lipid properties are important. pKa matters. Tail structure is important. Hydrophobicity, toxicity, all those things matter. When it comes to the payload, that has its own constraints. Sequence matters. Structure is important. chemical modifications, those things matter. At the particle level, everything depends on how the LNP assembles. Assembly conditions set particle size, RNA encapsulation, and internal structure. Once the particle enters the bloodstream, its behavior changes again. Proteins bind to the surface and form corona. That corona reshapes how the particle distributes in the body. And then at the cellular scale, delivery depends on escape from the endosome, a release of the payload, and efficient translation. So overall, the key point is this: all these variables are linked. You cannot understand or optimize them in isolation.